And that was actually a perfect lead-in because I think I have a great fear of overinterpretation of these very early studies in the combinations. I'm not going to present a lot of data and focus more on sort of where this combination field is going, what some of the upcoming targets are that will potentially be employed with combination therapies. So I'll review what we know of the two checkpoint inhibitors at a very high level and encourage everyone to wait for the very shortly forthcoming publications, which are probably more thoroughly reviewed and accurate than the abstract data currently available. Talk about new checkpoints and other immunomodulatory targets. And the chemo IO combos were already discussed, but just a brief word on them. So I hope you can see this all. This is a nice schema courtesy of Alex Schneider from one of her upcoming publications. That's been modified a bit, but talking sort of about the first and second signals of T-cell response. And I think there's been much more detailed overview of the complexities of the immune system already this morning. But just in really large, broad strokes, there's a tumor antigen, and based on a whole host of secondary signals and where it's presented, we have one of many downstream pathways that are triggered. And what are the secondary signals, both on the T cells and on the tumor cells? I'm not, for the most part, focusing on the microenvironment otherwise, that are potential therapeutic targets. And these are many, and there are more, but these are ones where we have drugs either here in testing or very soon to be tested. And as we know, CTLA-4, PD-1, and PD-L1, we have many drugs currently, well, I'm sorry, PD-L1s are not yet available, but close to available. So as I mentioned, I'll provide a very high-level overview of the combination data for lung cancer. This is the waterfall plot of the combination of durvalumab and tremelimumab in non-small cell lung cancer. It has a host of, it's a diverse patient population, including various lines of therapy. This will be very shortly. The manuscript is accepted for publication, so I'd refer you to that in the upcoming weeks to get into the details of the combination, as well as some nice, it was mentioned earlier that there hasn't been a lot of nice science provided for the rationale, and there's not a lot of predictive science, but there's a lot of good science that the company is working on to understand responses and the biology of their drugs in vivo. So durvalumab and tremelimumab does have substantial toxicity, but you can also see some nice responses in both PD-L1 and positive and PD-L1 negative patients. Same is true, and you've seen this already this morning for nivolumab and ipilimumab in the first-line setting. Very impressive responses, and what we've also seen presented on these data before, and like every other checkpoint inhibitor, when patients respond, the responses can be quite durable, and that's perhaps what's most exciting about these combos. So where, of course, when you don't have a lot of data, you get to show some pictures. This is a patient of mine on one of these combinations. I can't tell you which one, but she's had an absolutely remarkable response in her tumor, and this is a scan from month 13 on therapy. I could have updated it for later because she's still benefiting. The study schema has been brought up already. Dr. Carbone just outlined it, but the Checkmate 227 data was amended, or the study design, I'm sorry, was amended based on that very, very small sample size from their earlier multi-arm study. And now PD-L1 positive and negative are being treated differently to the extent that they were so worried about the poor response rate in the PD-L1 negative patients that the second combination of Ipi and Nivo, there were two different schedules being tested. That arm has now been amended to a chemo plus Nivo combo. So PD-L1 positive patients will receive nivolumab, Ipi, Nivo, or a chemo doublet, and PD-L1 negative patients will receive Ipi, Nivo, chemo plus Nivo, or a chemotherapy doublet. And that study is already accruing. The AstraZeneca counterparts have two studies in this space. The MYSTIC study was a first-line study collecting PD-L1 data, but not selecting by it. It's a combination of durvalumab and tremelimumab versus durvalumab versus the chemo doublet with a primary endpoint of DFS, and they have a Neptune study also in the launching process, if not already open, evaluating a standard care chemo doublet versus durvalumab and tremelimumab with an OS endpoint, and they believe that the data from these studies will be complementary. There's also a Pembro plus IPI study in development. I don't know how that collaboration came to be, but they've made it happen, so more data in that space. So where else are we going here? We know we have drugs to CTLA4 and PD1. There are multiple LAG3 inhibitors in clinical development. OX40, 2 drugs that I'm aware of. There may be others. I'm very entertained by the fact that the TRX518 drug is made by a company called Gitter Incorporated, so clearly this is a space moving very quickly. There are 2 drugs to CD137 in clinical practice, and CCR4 is a target I was previously unaware of. This is a drug approved in one of the hematologic malignancies now being evaluated in solid tumors, so clearly a very busy landscape. But there's more than just the T cell. We have IDO was mentioned this morning, or IDO. This drug I knew it by its letters and numbers. It now has a name that I can't pronounce. B7H3 is a new exciting target. We have the azacitidine class of molecules that have been studied both in combination and in sequence with checkpoint inhibitors. There are ongoing studies for safety. Where this will go, I know there's a lot of preclinical excitement. Where it will go clinically, I don't know. And I think that's one class or one area where predictive biomarkers will be very useful. Tumor-specific antigens, I mentioned that this morning, but how we'll use those in combination with checkpoint inhibitors is very an area with a lot of potential. I'll leave it at that. And then there are NK targets. There is a cure-directed drug in clinical practice, and there's a drug in development to NK2GA that's not yet in the clinic. And of course, we have all of the PD-L1 drugs available. So this was mentioned early, and then what I had planned to say on it has already been said in that last comment, is that, while everyone was jumping up and down about 80% response rates, 80% in a highly selected, very small population of patients. So we need to interpret the chemo-IO combinations with a grain of salt and look for the future. And again, I'll make a comment about sequencing. I think studying sequencing is happening more in advanced disease and of great interest. So what do we have to look forward to? I think short-term answers to the combination of CTLA4 and PD1 or L1. We have short-term answers to the chemo-IO combos. Intermediate-term answers about the safety and early efficacy signals of these new targets. A lot of the new targets are also being evaluated for Nivo or Pembro failures, understanding that's going to be an area of need in the future. And long-term questioning, sequencing, clearly predictive biomarkers to identify which combo for which patient. Thanks.
Just to start, so there are so many combinations out there, and for the drugs that work right now, our PD-L1 is just not a good biomarker. Are we going to, because the other biomarkers can be dynamic too, so how are we going to, do you have any thoughts, especially more that you guys may say, this is the way to predict for which combinations may work? I mean, any thought, is it going to be a platform, gene expression profile, any thought at all on how we should choose?
I think that we need a whole lot more work. I don't know that these drugs have moved so quickly into practice. So quickly into phase 3 studies, and everyone's so excited about getting their drug to the market first that I don't know there's been enough pre-planned analysis of these things. All of these phase 3s do require tissue, whether it's going to be enough tissue to do anything with.
Okay, so we had planned a discussion. We are now basically 15 minutes behind, but I think we're going to go take a break right now for 15 minutes and start at 10:45.
We'll start at 10:45. We'll have